You up on that. We had a great day at the Hilton. That was more in the morning. The working groups got together and met, and then in the afternoon, the working groups reported to the start of the full meeting, and that was more administrative. And today we get to have fun and actually do some science and everything like that. So that'll be great. We looked at governance and joining the Antibody Society, what that would look like. Excuse me. Looked at the relationship of the AIR community to important groups like IMGT or IUIS, the International Union of Immunological Societies. And as I said, had the working groups report on their progress over the last year and everything. Today we're concentrating more on scientific investigations, and then also we'll have the afternoon will be a session on IP and how that relates to distributed databases and AIR-seq databases. So anyway, I'd like to again welcome everybody. It's going to be a great meeting. We had an amazing international group, and that's why I'm a bit of a worrywart and got everybody over here early, but we got our international people through the security. One of the most important things is we have John Warren and Joe Breen, who've been so essential to this group and the building of this group, and they've provided us money and the venue, and so I'd like to everybody to give them a hand and appreciation of what they've done for us. And talking about getting everybody through security, Grace Tellini has been really a rock for getting all this done. So some more housekeeping things. We have a corrected booklet up on the air-community.org, AIRR-community.org. We have booklets here for everybody who wasn't here. You didn't pick it up yesterday. There's extra booklets. Posters, you can start putting your posters downstairs. We'll have a coffee break. You could go downstairs. We have a very short lunch today because the agenda is so packed, but that's the main time to look at the posters. And if you ordered your box lunch, those will be downstairs. We're not being really cheap. We don't have a huge amount of money for the meeting, but we can't actually order in catered coffee. So during the breaks or just go out, you have to purchase your coffee from the coffee stand there. Make sure you keep your name tags. Don't get rid of these name tags. You'll need them for tomorrow and Wednesday morning. I think that's about all the administrative things. So anyway, I'm going to turn it over to Andrew Collins and the morning session. Thank you.
On with the show. So those of you who were with us yesterday afternoon would have heard the report from the germline gene working group on the inference of alleles from AIR data. And this morning's session considers two utilities that aid that process of inference. In our presentation yesterday, we highlighted, I think there are 5 reported utilities for the inference of unreported polymorphisms to date. We're having presentations on 2. We have representatives of the developers of 3 of those utilities that I'm aware of. And the working group has been at pains to say that we actually, all of the utilities that we've examined are good at the task. The session here today is to bring those utilities to your attention, not to play one off against the other, just as, and I'm saying this actually because those who are not able to come up and present on their utilities, well, I hope they might have an opportunity to join us if there is anyone here in the panel discussion at 10 o'clock. But anyway, we should move on, and so I welcome Steve Kleinstein to give the first presentation on Tigger. Martin Corcoran, who will describe a very different approach to gene inference, describing IGDiscover.
Okay, thanks. I'd just like to start by thanking the organizers for allowing me to present some of our latest results. My name is Martin Corcoran, and I'm based in the group of Gunilla Carlsson Hedestam in Karolinska Institute in Stockholm. We're just about to move into a shiny new department called Biomedicum, and the very smart people here should take particular note of the triangular shaped building to the left there, because if you get a call from the Nobel Committee, that's where you go to give your acceptance speech. So I'm going to talk today about the basis for IGDiscover identification of IG germlines. And I'm going to talk extensively about various different validations we put in place to ensure that we're getting accurate germline output. Okay, so Steve has given a very nice introduction to the principles of germline identification. And there's a number of groups working with different algorithms to try to define this important process. Tigger, IGDiscover, Partis, Impre. And essentially what we're trying to do is the same thing. We're trying to isolate sequences which are used in multiple independent antibody rearrangements and which are present in the germline of the individuals. Now, there are two major inferral approaches, one which involves looking at sequences where there may not be any germline sequence at all present within it in an unmutated way. So, for example, a highly mutated data set may contain no examples of your actual germline sequence present. So you have to do an inferral process to figure out exactly what the germline sequence is. You do consensus building from that sequence. If you work from libraries which come from more naive sources, which we tend to do, you don't really have that type of problem because you can have large numbers of the germline sequences which are present already within the library. And just to try to give you a example of what I'm talking about, it's a bit like the where's Waldo question. So if you're working with a highly mutated thing, there may be very few examples of the real target present within your sequence. However, we're working with a more naive library where a substantial fraction of the sequences are there. So our task is a little bit different. We are simply trying to filter away mutated sequence, so we are left with the unmutated Waldos. Okay, so that might seem a little hyperbolic, in terms of the numbers there, but it's not really, because if you actually look at the number of, this is an output from IGDiscover, if you look at the number of exact sequences present in an IgG library and an IgM library, you'll notice that you get about 1 to 1.5% of unmutated germline sequences present in the IgG library, whereas in the IgM libraries that we're working with, we get something like over 20%. So we have a significant target to actually filter out. So the basic process for IGDiscover, it was originally developed actually not to work with human sequences, but we're working a lot with rhesus libraries. And the available databases were not sufficiently comprehensive enough for us to be sure we were getting accurate assignments. So we tried to develop a process where we could use the expressed sequences to infer the rhesus germlines. And the basic process involved doing MySeq libraries 2x300 base pair. And then we did assignment using the IGBLAST module, and then we looked for clusters of sequences which were assigned to the what we call the starting database genes. So each one of the genes where we had assignment to, we split them up into 2% windows and looked for clusters of sequences within those. So to do that, we derived consensus sequences from each of the 2% windows. And those consensus sequences we searched through the raw data and then parsed out the sequences which corresponded to that. We also developed another cluster identification process which took all the sequences which are assigned to a single gene and then compared them to each other. And we were able to derive consensus sequences from those, and we also searched the original sequence for that. So that allowed us to actually identify what we call candidate sequences. So these are large numbers of sequences which contained germline sequences plus a large number of false positives. So we developed various filtration methods to remove the false positives and leave behind higher quality germline candidates. Now, when we actually do IGDiscover, we originally did it in an iterative manner. So because we know that in rhesus there are quite a number of genes which are not present in the current germline databases, we realized that a single iteration of IGDiscover would likely just find the highly expressed genes, but we wouldn't be able to find the low expressed genes. So we developed what's called a database replacement model, which means that when we do our initial analysis and of the sequences, we create a new database, and we subsequently use that database in the next round. So we don't assume that any single starting gene, which is in the reference database, is present within it. So any gene could be replaced. And we do this in an iterative manner. When we did it originally, we were working up to 14 iterations. For human now, we're down to one single iteration. And for rhesus, I think 2 or 3 iterations gives us all the genes. So when we do cluster analysis, this is an example of the candidate germline sequences. You may not be able to see this very well, but this is an example of IGH 1801. So if you look at the results we get when we parse out the sequences, we look at all the associated markers for germline sequences. And these are particular factors which we have noticed are highly associated with sequences which are rearranged multiple times. So that could be numbers of unique CDR3s, number of J's used, number of D's used. The D thing is something we recently introduced into IGDiscover, and that's actually been very helpful for removing potential false positives. It's very useful to actually include D assignment as well. So in this way, we identify candidate germline sequences, and then we parse the data using the germline filter, looking for sequences which have multiple CDR3 usage, multiple J usage, multiple D usage, and that's essentially part of our germline filter. And we also do something which is called the allelic ratio. And the allelic ratio is something we noticed when we sort of examine the candidate sequences. And what we notice is that the false positive sequences tend to be relatively frequent, but at a rate which is very low compared to real germline sequences. So what that means is that you're able to use the ratio between the germline sequence you identify to exclude false positives. This is just an example of a series of heterozygous alleles from one individual. And if you examine the ratio between the highest expressed allele of each allele pair, the gene, you will notice that there is an allele ratio which is, in this case, all above 0.31. And in our germline filtration step, we use a allele ratio of 0.1. So it seems to be that the false positives tend to be around about the 1% level. So if you set it a relatively conservative allele ratio of about 0.1, you will remove almost all the false positives. So when we do this cluster identification and looking at the numbers of J's and D's and CDR3 usage, that's it's quite useful to find high expressed genes, but low expressed genes are a problem. And if we want to get a full genotype for an individual, we will want to get both the high expressed plus the low expressed genes. So we developed a technique called whitelisting, which means that we use a lower cluster number, but we still need to have multiple CDR3 and J usage in those cases. And that allows us to actually get very, very low expressed genes. And we actually thought that this actually might make a useful test just to demonstrate the levels of false positives that you get in individuals. So we took one gene, 11801 again, and we did single nucleotide changes to create single base changes, every possible single base change of that reference gene. So this reference gene, we know was expressed in that individual. So we created a 1780 sequences of that. So every possible single base change has been added to the database. And what that means is the whitelist method in IGDiscover can pull in every sequence which corresponds to these. So as long as they are present at least 5 times, it will pull them into the output. And that allows us to actually see where the false positives are possibly coming in, because I will pull in every single example of this. So on the left-hand side there, you just see the 1801, which is not mutated, and we're getting about 1,500 unique CDR3s in that. And you can see that right across the gene, you're not getting very high numbers of other sequences assigned. Until you come to the very end, the last 2 codons is where you actually get significant possible false positives coming up. So that's one thing you have to be a little bit careful about. So if you can use the allelic ratio, but the last 2 codons, you have to be a little bit careful. But even still, they're usually below the 0.1 cutoff level. We tried that in another gene, and we actually noticed that one sequence actually came up. But that turns out that is a single nucleotide difference from 16901 is actually 16906, which that individual had. That individual had 3 alleles of 169. And we did confirm this by haplotype. I don't think I have the picture now, but maybe Nilla will show it tomorrow. So the process of iteration, I mentioned we sort of developed it for the rhesus question. And this allows us to actually go from very small numbers of starting genes in the database, and in an iterative fashion replace the database each time until you end up with a full genotype for the individual. And we can use this process to sort of test out the program. Because if we have a full genotype for an individual, we can see what types of changes in the starting database will still allow us to actually find genes. So for example, if we use the IMGT database, we find all 61 alleles in a single iteration. If we mutate every one of the IMGT alleles by a single nucleotide randomly, we find 57 out of 61, which basically means we find all apart from a small number of low expressed sequences. And interesting, if you keep doing the mutations, if you increase 5 mutations, 10 mutations, or even 100 mutations per sequence, you still find the same number, although you have to do extra iterations. You can actually use the rhesus database to get a full human genotype. So it replaces the rhesus sequences with the human sequences it finds. It simply uses the rhesus sequences as comparison points. And then it derives the clusters from those comparisons. And we've even done some extreme tests as well. We used a single dolphin gene or a single rhinoceros gene, and a single zebrafish gene. You don't quite get all the human sequences in this way, but it's simply to test the ability of the program to replace the database in this way. And we want to use this in future to identify germline databases in animals where the reference sequence is not available. Okay, so on to some validation. So one validation thing that we were very interested in doing was to analyze the expressed genotype from an individual where the germline genomic sequence was available. And we looked around for quite a while to see if anybody had a full genomic sequence of the IG locus and also had expressed libraries available. We weren't able to find anyone. So that would have been the best test, but we thought another test would be simply to do full genotyping from an individual from whom we had already identified a database. So what we did was we targeted every single VH gene. We designed specific primers upstream of the L1 exon and downstream of the L2 exon. In order to get this to work, we had to design specific primers for each gene. Using family-specific primers was not sufficient. We did Sanger sequencing of these, and we were able to validate all 61 alleles from these, which about 50 individual genes. So we were thinking that it would be very good in future that other genotypes were available, so that if additional tools for germline inference were becoming available, we could work with the same type of data set. So we will make available these data sets for people to test with other tools when we are publishing our study. Another thing we've been doing is validating the germline sequences that we've identified by finding them in more than one individual and in other databases which people have deposited novel alleles in the past. So we've identified 128 novel alleles in total. 45 of these were present in more than one individual. 7 novel alleles were found in more than 10 individuals. 2 of them were found in more than 30 individuals out of a total of 66 cases. We found from the 128 alleles, 44 of them were present in the IGPDB. So these are sequences that people have deposited years past. So I think it's quite useful to actually have these various sources that we can do these comparisons. We've done validation using haplotype analysis. Matt Solin, who will be talking directly after me, had a very interesting study published earlier this year where he was able to use IGDiscover to look at haplotypes in individuals that he was working on. So essentially what that involves, and I won't talk too much about this, is using the association between polymorphic J alleles to identify on which chromosome the either maternal or paternal chromosome the V genes are situated on. And we have developed a module within IGDiscover to actually do this, so we can actually have very nice haplotype outputs from heterozygous individuals as part of IGDiscover at the moment. And this can be very useful for validating novel alleles. For example, this particular allele here, it's a variant of V32001, nicely segregates on the J602 associated chromosome and the J603 chromosome. So this is one particular method that I think will be very useful in future. We've also been using validation using familial data. We've looked at monozygous twin data. This is not data that we produced ourselves. We've taken the raw data from Florian Rubelt's study from last year. They had deposited some data in SRA, so we were able to retrieve that data. With 5 sets of monozygous twins, we were able to extract the IgM sequences and analyze them for IGDiscover. Out of those 5 pairs of twins, 4 of them had concordant novel alleles. So pair 2 had a variant of 270, pair 3 of 439. Twin pair 4 had V12P06, which is an allele that has been discussed much within the germline group. You won't be able to see this very well, so I've expanded it up a little bit here. You would expect, because the germline content of people tends to be almost like a fingerprint for an individual, that you should get from twins. You should be able to get a corresponding output. And we got very nice results. There was only like 2 or 3 alleles which didn't come up, but they were also present within the candidate list, so they were just below the level of detection. And we think that was because there wasn't sufficient sequences overall in the IgM library. There was 4 novel alleles present within those twins. 3 of them had examples within our other 128 sequences, and 1 of them was completely novel. And also we looked at inheritance of a novel allele. So we had 2 individuals, 1 a parent and 1 a child, and those 2 individuals shared a novel allele. Now, out of 66 individuals we've analyzed, only 2 individuals had this particular novel allele, and they were the parent and child here. And we were able to show that the various alleles segregated in a nice Mendelian fashion, apart from the alleles for VH7401, 7402, which seemed to be discordant for some reason. However, haplotype analysis indicated that the reason they were discordant was the mother had a deletion of VH7401, and she passed that deletion to the child. The novel allele segregates out on the mother's chromosome as well. We started a human diversity study looking at different human groups. I'm not going to talk about this now. Nilla will go into detail tomorrow. And we have done some comparison in terms of what we see in the IMGT database and how frequently we see known genes appear. So just to summarize, we think that germlines can be identified with high degrees of confidence because there are multiple validation methods that we can apply to these. And one important thing I should just finally say is that individualized databases makes things a lot simpler. So just to see the sequence of one novel gene on its own is not really enough. You really need to have like the sequence from or the output for the full genotype to actually give you a better picture of whether you can trust those sequences or not. Just finally, just to acknowledge my research group. I work in the Gunilla Carlson-Hedison group as part of the NGS and non-human primate team with colleagues Ganesh, Nestor, Sanjana, Paula, who's just left us, and Pradeepa. And we have been doing some work also with Catherine Sheepers, Lynn Morris, and Penny Moore. And I think Catherine has a nice poster today. And Gunilla will show some of the data from the Caprisa study that we've been helping with tomorrow. Okay, thank you.
Thank you, Martin. Do we have questions? Can you please come to the mic?
It's fantastic work. I'm wondering if there is a relatively simple modification that you can make that would allow it to be applied to a large mixed database. So obviously most of our data that comes in is person by person, but if you wanted to just throw all IG sequences that are in SRA or all IG sequences that are in the broader NIH databases, excuse me, to just say, okay, what are all the alleles that one can discover? Is there a simple modification that would allow this to be done?
There might be. I can't think of one. The trouble is that we tend to use an allelic ratio as our majority filter.
Yes.
So when you have multiple individuals, you can't really do that. So yeah, sorry about that.
So your algorithm works so well on naive data sets, but how naive the data set should be for the method to work? Presumably at some level of mutations, it will break, and it looks like for 5 sets of twins, there are actually data sets that have quite a bit of mutations, making it difficult to apply your method.
Okay, I actually had some slides that dealt exactly with that. I'm not quite sure if I can't go forward. Oops. Yeah, so this is the slides that go with that. So I showed originally some data showing the differences in number of sequences, exact sequences within an IgM library compared to an IgG library. So we did a test where we mixed the two libraries together. And in that case, you drop the number of IgM sequences by half or so, approximately. They're approximately equal size libraries to begin with. And if I can, yeah, the result for that is that we were able to identify 52 out of 53 alleles that were present in the first one. We didn't get additional false positives. We just lost one gene. We did that, but it was very close to the cutoff. And it could be that it's simply a question of adjusting the config file, adjusting the cutoffs to allow you to do that. So we have a lot of parameters which are adjustable within IGDiscover. So you can do these various set up the cutoffs slightly different, and it may allow you to do this. So as I've shown there, you can have a mixed IgG, IgM library, and it still essentially finds the same thing as you find in the IgM library.
Just add a comment to that. We often get this question, and of course, when you talk about naive starting samples, we have used samples that are pre-immunization, post-immunization, peak responses. I mean, that they all give the same output if you use an IgM library. So, it doesn't really matter if the individual is, young, old, you get the same.
So, is it correct then, or would you say it's correct that really the best standard for the alignment would be to have the germline alleles? So, for example, if you're doing an experiment with 20 outbred animals, why not just capture the germline alleles along with any of the recombinations you want to align? Or if you're doing a prospective clinical trial, prospective human study, is that the highest standard to capture the germline versions? And
well, I think that's the model that we're working with. Yeah, we're working with Rhesus, and when we're doing the immunization studies at the moment, we create an individualized database for each monkey that we're doing the studies on, and we're finding very interesting antibodies, which we can identify the germlines, and these are novel germlines, so we wouldn't be able to tell develop good lineage trees without knowing the base of the tree. So in IGDiscover, we can identify novel J's as well, and very recently we figured out how to identify novel D's, and we're integrating that into the program as well. So I think we would sort of envisage in the future that people will be able to look at the individualized VD and J.
So it would also be your sense in a clinical trial, if you had, say, 100 patients, just capture the germline alleles and use on a case-by-case basis those alleles to align.
Exactly. I think that is the way forward.
Final question, Uri.
So I was just wondering, when you showed us those positions that they did confuse the alleles at the end of the V. So those are the last 6?
Yeah, there's the last 2 codons.
So those are nearly identical in all V's. Couldn't you just put some parameter there that said, well, those are just always the same, so we shouldn't use those?
Well, those are the false positives which you see in the data, but they are still below the cutoff level. So when you actually get the output for IGDiscover, you don't get false positives at the end. Sometimes you get a sequence which is 1 or 2 bases too short, but you don't get.
No, but I was just wondering if you could sort of. I think maybe the problem you're having is that those positions are the least different amongst alleles of all. They're always the same. So maybe your algorithm is sort of not. And you could maybe tell it explicitly. Those positions don't change.
Yeah. I think the problem of the last 2 codons is one that people have had for quite a while, and I think most assignment programs they take that into account that you cannot be confident with the last few nucleotides.
In fact, I think we might move on and hear more about those final nucleotides from Matt Zolan, who will talk further on evaluation and validation. Thanks, Matt.
Good. Okay, thank you everyone, and thank you Felix and Jamie for inviting me to this session. So I'm not a computational biologist. I'm interested in biology of the immune system, and so I've taken a very practical approach to this matter, and that's really so much of what I'm going to talk to you about today about having valid results being studied in immune or antibody research is from this particular slide. So this was pre-Tigger era, and we were studying the mutation frequency in IgG populations in bone marrow of subjects, and in general, you saw very so essentially every codon different or evolved very similarly in different subjects. But in this particular study, we had identified individuals that we thought were homozygous for 1,2. That has been discussed previously today. But it turned out that in one codon, there was a very large standard deviation in the degree of perceived mutations. So we needed to look further into this matter. So in IGT, there are 5 alleles of this gene, all of which are very similar, and that, of course, evokes problems in the analysis. So if you look in the unmutated or very slightly mutated V gene calls of these three individuals, just as an example, you would see some that are perceived as homozygous for 1,2, and others are heterozygous for 1,2 alleles. But in some individuals, you will see a lot of undecided calls. And these are the calls in these 3 individuals that caused the havoc in our analysis, because these really represent an allele that is not recognized by IMGT. Also, when you look at IgG repertoires that are highly mutated, for instance, this is just exemplified here with 348. You will have, in the IgM population, you will see nice descriptions of the allele calls. In this case, either 1,2 or 3,4 in these individuals. But if you look in the IgG calls, you will see a lot of misrepresentations that amount to quite large fractions of the entire population of genes. So that will also create a lot of misinterpretation of mutational effects. So we need to go into working with individualized germline databases to actually resolve this matter. And of course, in this process, we need to work with a process that gives us confidence in the calls that we're going to make. So we need high specificity and high sensitivity of the detection system. And we have turned then to Tigger and mostly to IGDiscover that emerged on the market around this time. But there are, of course, many other softwares available. But the principles that I'm going to talk about now are probably going to be similar. So if we look on some of the more problematic genes that we have experienced, so 1,2 is one, but also 3,7, and we work with Tigger, it seems So we're working mostly with IgM populations, but they contain both mutated and unmutated sequences. But the software has some problems in actually defining whether these individuals are homozygous or heterozygous. And to some extent, this is also settings dependent. So it's very important how you set the analysis parameters. And there are many parameters to set in these tests. And I'm not certain that all users will be sort of fully appreciative of this fact. So, and also when we turn to IGDiscover, and we use, if we use as a starting database IMGT containing the normal the normal alleles of 1.2 and set a specific parameter, the difference parameter to 0, we will detect a set of allele calls that we, through a lot of downstream analysis, believe are valid. However, if we were to replace these 5 alleles of 1, 2 with the allele of the mutated version of allele that both Martin and Steve has discussed, we would also get the same output using the difference 0 parameter setting. However, if we were to use other settings, we would get other results. And at that time, the difference parameter was set at 1, I believe, as the default in the software. But we're happy now that parameter has changed over time, and because it provided a lack of specificity, but also a lack of sensitivity of the assay. And of course, if you had a different starting database, you would perceive, if you used other settings of the difference parameter, you would perceive that you actually had other germline genes in your population. So often these inference technologies are also there to try to discover new germline genes. Also, if you have very limited knowledge of the germline repertoire. So we have used IGDiscover to try to discover the repertoires based on databases that were IMGT as it stands, or using very small sets of V genes, either from 3 germline subgroups, and two such test sets. And we will discuss, so some germline genes will be discovered independently of the database that we're using. But some other germline genes may only be discovered if there is a very large set of germline genes in your starting database. And others will be discovered only if you have a close, similar germline gene in your database. So for instance, unless you have a germline gene, so 118 in the database, you will, only if that's the case, you will detect this 1.2. And similarly, 459 will only be detected if there is a 4 subgroup member in the database. So that said, I think these tools work very, very nicely to discover a lot of the germline genes of these individuals. So here are some examples. We have been studying, in this case, bone marrow of donors, and we can discover also new alleles, in this case of this 1, 2, but we have other examples as well. And many of course of the well-defined genes, they get good calls by this approach. And I will describe later on how we validate these calls further. So here is another donor, and we have a different set of calls. And it doesn't really matter if we study bone marrow, or if we study peripheral blood, you get very much from the same individual. You get the same kind of calls. But there are some other calls that, as we'll see downstream, they are more problematic. So here is a call of 438.2, where we see an allelic variant at a quite appreciable level. So it's about 15% or so of the calls of the. So this is the allelic variant of the IMGT recognized gene. So and for all of these calls that we get, we want to make sure that they are true. So what we developed, and these were found both in peripheral blood and bone marrow. So there was no discordance in the data. It was reproducible finding. So what we developed was this haplotype analysis system. So we, starting from the idea that we have 2 chromosomes, if we are to discover more than one allele of a single gene, they would have to be distributed across the 2 chromosomes, of course, or represent copy number variations. And they could be linked either to chromosomal differences in J gene representations, or in like allele 2 or 3 that Martin described previously, or they could be linked to differences in representations of the D alleles, or in the presence of the or in the presence or not of certain D genes. A thing that Andrew described and Catherine Jackson last year. So let's say that we have this particular output. So we will see, for instance, the gene 2,5 would be associated to both of the alleles of the J6. This would be found on both chromosomes. And if we had different alleles, so 1, 2, they might distribute differently between different alleles of J6, or it may distribute differently on the D alleles. However, if we had this kind of call, that is that 2 allelic variants were associated with one specific J gene only, that would be problematic and call for some further validation. So this is real data that we get out from this kind of assay, similar to what Martin showed previously. So here we see, so we have assayed these samples both in peripheral blood and bone marrow. And so we link all the gene calls to the alleles of J6, and we get very similar results in peripheral blood and bone marrow. So for instance, here is a typical call that one can have some confidence in. So we have one call of 43D01, and that's distributed to both J alleles. And we have 2 calls of 348, alleles 3 and 4, and they segregate between the 2 different J genes, which seems similar, reasonable. Also, in the new discovered, or the new allele that has been described by Scott Boyd several years ago and then further discussed by Steve and Martin, they also segregate very well between the haplotypes as defined by the J segments, and that's very reproducible. However, when we come to this problematic gene 438.2 that we saw, it turns out that this the previously known allele was present on both haplotypes, and also the new discovered allele seemed to be present similarly on both haplotypes. So that would imply that it's inferred incorrectly, or that it requires some copy number variation. Similarly, with allele calls that were made with 439, in this particular individual was only on one of the haplotypes. All of the incorrect or all of the calls that were questionable were on that same haplotype, suggesting that they are either made in error or that they represent the copy number variation. So we also look on so we go back to the original FASTIQ files and pull out the reads and the quality associated with all of the reads, and we look for how well are the what's the quality of the data that underlies these inferences, and we are looking here specifically at the allele differentiating calls. And as you can see, for these two new alleles, there is very good quality, not only in the previously known alleles, but also for the reads that represent the new call. So that there is not a technical artifact in terms of sequencing associated to this particular call. However, with some other calls, we see a substantial drop in the quality of the reads of those reads that represent that call. But it's not a general bad quality of those reads. It's a local problem associated with a specific base that appears to be sensitive to sequencing errors in the MySeq platform that we're using. So certainly there are multiple evidence based on these findings that suggest that these calls should not be considered. And this actually aids all of us in our efforts to develop good confidence in the kind of personalized databases that we build, or which calls that should actually be included in those databases. So I will also turn a little to what what Martin was discussing, and that was the 3' end problem of the V gene, because the 3' end is associated with a enzymatic process that may or may not remove bases before or during the recombination process. And this may impact on inference. So for instance, if we have this, if we infer using the entire sequence of the database of IMGT, because the data I showed previously, we had restricted our inference to only go up to codon 105, so the codon after the conserved cysteine. But if we had the entire IMGT database, we might see inferences like this mutation in codon 106, and at quite appreciable frequencies. So it may well be above the cutoff that is normally used, like 10% or 12.5%, as was discussed before. So can we have confidence in this? So we used haplotype analysis again, and in this particular donor, we have for 348, they segregate very well between the two haplotypes. But when we look into 118, we see that the variant also segregates exactly like the previously known gene. So again, implying that it's made in error or that it's requires a copy number variation. And indeed, there are many of these calls that you can identify that reach quite appreciable levels. So this implies that there is a problem if we want to use inference to reach very far into the V gene by itself. So probably we need to apply other standards when we move beyond codon 105, and that was sort of similar to what you were implying earlier. So we were looking in a in several individuals on how which bases are actually present at different positions after the cysteines, which is over here. So this is codon 106, and in all cases, almost, we get precisely the codon that is represented in the IMGT database. But the second most common base may be more than 15% of the calls, so that is certainly going to be a problem. Something we need to consider. There is one case we see, and that's an allele of 37. So in 372, those genes that are called as 372, we almost always see a G, despite the fact that this is supposedly in the IMGT database going to be an A. And in the other allele of 37, we see the expected A. Now, this particular allele, 372, has not been sequenced beyond this particular base, and is not described by IMGT beyond this particular base. So we cannot really know if the enzyme modification or the trimming of the gene has proceeded all this way and preferentially incorporate the G. But the GA sitting in these two bases are similar to what you would expect to see, and you also see those in 371. So there is it's not unlikely that this is a true variant of 372, and we have seen this in several individuals. So I think this is based on 3 individuals, and we've also looked in other database in other data sets, and we see the same kind of variant. So when we move further on, because we would like to move as far down the gene as possible, so also in base 319, so the second last base of most V genes, we see again the V gene, or the IMGT base being the most predominant. But certainly other bases reach well above 15%. So we need to apply other standards also for that base. So then we come to the last base of the V gene, and for many genes, it appears appropriately in relation to what's in the IMGT, which in these cases would be an A. But in others, it becomes more difficult, because would you like to call this an A? And what's the evidence? Or would you have a preformed impression of what should be there based on what is in the germline database? And this is even worse. So would you expect this to be an A? No. It should be according to... So my point here is that as we move towards the end of the V gene, there is not really a possibility to infer the V gene. So we need to apply other rules as we approach the end of the gene. And in this particular case, it actually has some impact also on the protein, because whether there is an A or not is going to make a big difference in the protein product that's going to be made, because the GA sequence implies that there's going to be a negatively charged amino acid, but in these particular amino acid genes, that it's going to be a glycine instead, and that's going to impact on what's available in the repertoire of these coming out from these V genes. So in summary, I think that we can be assured that gene inference provide very good quality data that we can in many cases trust, but also that and that's going to be important for valid downstream research, because we are going to be able to use the individual's germline genes and not make the wrong calls in our research studies. However, it's important to be aware of that the starting germline gene library or parameter settings are going to impact or may impact your output depending on how you run your assay. And I think in that sense, many users may not be so sort of, they are more users than actually down to earth of the details of the programs and what it actually does. So, so it's important that in research studies that you actually report properly all of the settings that you have been using in your studies, and also that when we evaluate the quality or the relevance of germline gene calls, that this is known to the group that's going to make these calls. Or I also think, and I agree with Martin, that haplotype analysis adds confidence to the germline gene calls beyond what's there in just computational analysis of frequencies. And it also identifies those calls that are in need for further evaluation downstream. And also that germline, that read quality analysis adds additional levels of confidence to the gene calls. And finally, that we need to apply stricter criteria as we move towards the end of the gene. So thank you for your attention. Actually, maybe I should also mention the people that have done much of the work. So it's Ufuk Kirik, who was a postdoc bioinformatician in my group, and Linnea Törnqvist, who has done much of the work on the 3' end study. Matthias Levin, while he was in my group, he collected the material that we have used for these studies as part of his studies of IG responses and studies of immune repertoires in bone marrow and peripheral blood. So, and I'm also indebted to Scott Boyd and his group for collaborating a lot on immunoglobulin and really putting us on the market in terms of next generation sequencing analysis. So thank you very much.
Thanks, Mats. Do we have any questions? Yes.
Just one question is, when you're looking at the V genes and there's like perhaps a different length V gene, everything is the same before the 104, but the edge nucleotide has been deleted off. Like, could you use that method to say, okay, this is probably a different V gene with a different length?
Or if you,
because there are some like V genes that are very similar, but the number of nucleotides following the 104 codon differ. Some are deleted off.
Yeah.
And so, could you use your method to say this is a new V gene, or?
Yeah, I would say.
And I guess the other question is when we're doing these allele discovery, has anyone like actually started using like somatic hypermutation substitution patterns to increase the confidence of a new allele? Say that this gene keeps on showing that it's a T to C mutation, which doesn't make sense with the mechanisms of somatic hypermutation, so it's probably a new allele. Like, has that been factored in to increase the confidence of a allele discovery?
Well, I mean, that was sort of the starting incentive for us to do the work. But I wouldn't say that anyone had have done much work on defining mutational patterns in different alleles. There has been work on defining germline specific mutation patterns, both by us and by Schramm. I haven't seen him here. so there are certainly so mutational patterns are certainly important in and related to the germline gene origin.
I'm curious about the codons at the end of the V segment that you were just referring to.
Yeah.
So in the case where you're changing positive charge to like glycine, for example, is that global in these individuals, or is it just the diversity in the repertoire at that position? And I'm asking the question because whether or not that infers something about the structure of the antibody. So I'm trying to link that boundary of the CDR3 to something.
Yeah.
And I'm just trying to understand a little bit more about what you're thinking about that.
Yeah, so that's a very good question. So the side chain of polar amino acids at position 107 that this is about, it often makes or there are potential hydrogen bonds being made with side chains of codon of amino acid 40, which is the first amino acid after CDR1. And of course, exchanging that for something else, it may have substantial impact on the structure of the antibody or the positioning of the hypervariable loop. But this is something that appears to occur. I mean, this happens already at the stage of rearrangements. Whether that also implies, so is this a way that you can evolve the antibodies also further on by somatic hypermutation? It's currently not known, and I mean, one thing that might be very interesting would, of course, be to do mutagenesis studies and structural investigations, and what these changes imply in terms of loop structure. But it has not been done yet.
Thanks, Mats. If there are no further questions, I'd like to thank Mats for his talk, and I'd actually now like to ask the previous two speakers to join us here for a group discussion, and I'd also like to ask Duncan Ralph to join us. Duncan has just posted a preprint of an evaluation of IGDiscover and Tigger as well as Partis. And in fact, we might begin if I can put you on the spot. Would you perhaps just like to tell us a little bit about your preprint?
Sure. Should I stand up? Yeah. I hope that made it clear that I did not prepare this. Let's see. Yeah, so we just got out a preprint about 2 weeks ago. It's on archive. You can log on Eric's Twitter feed is maybe the easiest way to find it, or just ask me for a link. It's definitely very strongly influenced by IGDiscover and Tigger. There were just, can you hear me? It's not close enough. Is that better? Thanks. Let's see. There were just some things we wanted to improve a little bit on it, and it's just always good to have a lot of different diversity. As far as more specifics, that'd be great to talk about in the session, or I also have a poster up. Anything else?
That's fine. Okay. So, yeah, perhaps I should mention the recommendations of the germline working group that was presented yesterday, and in fact, there's a document that will be available to you all, presenting the recommendations, and they'll be voted on Wednesday. And one of the issues that we, in fact, discussed was the issue of the 3' end of inferred sequences. And in fact, there are two issues here. We don't want to take the inference too far and get into trouble, but at the same time, we have in the past had short genes in the database, including inferred incomplete genes in the database, and we no longer want to see that happen. So one of the recommendations that we made is that for inferred sequences to be accepted, they should be complete to the end of codon 105, but we make no recommendations beyond codon 105. Would anyone like to comment? Yeah, Murray Paul. Oh, would you mind speaking at the mic?
The limit of the 3' V region, in fact, is just the beginning of the VRS, of the heptamer. I think it can be, theoretically, if you have germline genes, we cut the 3' V region when the heptamer, which fits, in fact, what has happened in the rearrangement. So I think we do not check about the number of the codon, we check at the nucleotide level, and it may have an even number because you just cut at the heptamer. So this is the official limit. And what we are trying to do, for instance, we realize that many old allele and have never been fully sequenced. But you remember in your work, for instance, when you have new alleles, we corrected many, it was, I think, a substantial number of 3' V region just to complete. I say, oh, this sequence is interesting. It brings maybe only 1 or 2 nucleotides, but at least we knew that it was improving the junction analysis. So I think even if you come through genomic genes already known, I think really it will be great for the community that to check if the sequence is complete in 3', which is very easy to see because usually the first allele is complete and the other allele are, we can see that there is a suspect. So it's very, very easy to check. Just have a look at the alignment of allele. And if you bring 1 or 2 nucleotides, please come. It's very welcome.
Are there questions anyone would like to throw to the panel? Uri, thank you. The two Uris.
Two Uris. So we'll do it one after the other. But so I was just wondering, in some of the examples that you showed, so by looking at the population, you could tell that they had a certain amount of alleles and that you used that for your confidence. But sometimes even genes are very similar to each other. So like all the J genes, definitely all the 6 genes, alleles, they're essentially identical, especially if you have some level of mutation. And even IgMs do mutate. So I was a little bit, I didn't completely understand how you could tell with confidence which one was which, consistently enough that you weren't sort of mixing apples and oranges as you were making your model. So if you could talk about that a little bit, that would help.
So I think this is true for all the tools, that we really don't make a distinction between a gene and an allele. That what you can find from these data is that there is some, some segment that is different from what's in the databases and what's known. Whether or not that represents an allele of a gene that's already in there, or whether that's a novel gene, I don't think we can tell from this kind of analysis.
But doesn't that sort of destroy your assumptions about how many different copies of something you should have, different kinds you should have? So like, I don't know, there's a whole group of VH3 genes that are just one of them I think is 1 mutation apart. And if you don't actually, I mean, if you know that person's germline, then it's better. But if you don't, then I don't know how you can tell that's a new thing or just a mutated one. Or how do you know that you don't have another new thing, another that's just so similar to the other ones, and suddenly you have another allele that you can't identify because it looks like one of the original genes?
The answer to this may be that's how it shakes out in the math, and if you look, it's hard to describe just in front of a room.
Can you?
No, I think it's possible. Like you could have a known allele in a homozygous state that's allele 01, and you could have a duplication of that that's in a homozygous state in a novel allele, and that will look, excuse me, that will look like a heterozygous novel plus a, plus the germline. And I don't think any of the tools currently distinguish that situation. As one can try to potentially go into expression levels of those segments, but I think that's that's where we are. I don't think that that particular case is not something you can tell apart at this point with the with these kinds of methods. As far as anyone else wants to comment on that.
So we have a case with 459 where we in many individuals see two alleles. So 459.1, 459.1, and 459.8. Now in these individuals, it turns out that 459.1 is on both chromosomes. So what about 459.8? That would imply that it's a copy number variation. Now, in these individuals, there was a paper earlier this year that was studying some population in Oceania in relation to some disease. I don't remember which one it was. And they linked this to a new allele of 461 that was sequenced in the genome. This particular allele appeared to be, it was reported as a 461 allele, but it's identical to the sequence of 459.8. So potentially 459.8 is sitting in these individuals at 461. So in our data, how is the situation in 461? So in these individuals, we see that 461 is only found on the chromosome that does not hold 459.8. But of course, it's not proof.
Actually, I can confirm that we have haplotypes where there is no 461, but a 459 star 08 next to a star 01.
Okay, yeah.
So
pretty good.
We've also found an individual who has 459, 01, and 08 on two chromosomes. So it sort of confirms that, yeah, you have this type of duplication going on or additional allele.
Ulrich?
So what would happen if, so Steve, you say how you have a cutoff of like 30 mutations potentially, then you can start with whatever database you want iteratively.
We don't have a cutoff. It's arbitrary. We test it up to 30.
So what would happen if, for example, in the starting database, you use the entire human genome? Would you be able to find these like weird layer 1 antibodies in people's data sets?
I don't know. Actually, we should talk about that. Well, I mean, we could actually start off with nothing known by sort of seeding. One way we've run it is by sort of doing consensus and seeding with those consensus sequences and finding from there. So in theory, you could find sort of arbitrary genes.
So there were great new results on this segment discovery presented today. But with all due respect, I don't see it as a new algorithmic problem. Because let's don't forget that these segments are just repeats. And people invested in bioinformatics, invested a lot of efforts into repeat subfamily classification. It's an old problem, 20 years old problem with sophisticated methods. And somehow this community doesn't look into this old and still developing studies. And I wonder whether you see a possibility to finally bring these two areas together. And in this area, like when you look at 1 million of RepSec reads, think about them as 1 million of allele repeats in human genome. And these allele repeats were derived with mutation from 100-something subfamilies. So deriving de novo these subfamilies, allele subfamilies, from million allele repeats is absolutely, algorithmically, is the same problem. So I wonder why this community, and there is a possibility, for this community actually to bring these two directions together, because for repeat subfamily classification, for example, there are p-values and there is a lot of work done, and I hope that it can actually contribute to what is done for V-segment classifications. They are just repeats.
Yeah, I mean, at least from what I know of the difference, I'd love to talk about it more. I mean, the difficulty starts to come with the somatic hypermutation patterns, which are sort of a very high fraction of the bases, and particularly show these intrinsic biases. So it's not stochastic patterns, but you have very particular positions which can have the same exact point substitution in a very high percentage of the sequences, and that's one of the things that makes, as far as I know, makes this situation particularly unique. But I'd love to discuss it.
Pavel's question made me think maybe we should have had the germline session first and then the inferred, because that's what we'll be talking about in the next session. And there have been, I think Corey's really worked a lot on, trying to sequence through these really difficult regions, and then several people have tried to just algorithmically construct these haplotypes, because they're, like you say, they're just a bunch of repeats. But it's gone back and forth in the community how well these actual haplotypes have been reconstructed through these algorithmic approaches. So people have tried to do it. There's been a lot of discussion whether it's possible and how well, and that's one of the things we'll be looking at in the next section.
As far as inferring new V genes, like the ones that you were just talking about in the last talk, if you've got a good idea, you've got individuals identified that most likely have this, do you then sequence up the germline DNA and just definitively show that that is the germline gene? Because we have done that to identify some new germline genes, just PCR, clone, and sequence and show that it's in the genome, and then you can put it in the database as a known gene.
For many studies of these repertoires, there may not be access to the patient if you're using third-party data, or if you're with the patients that we have been studying, we have no permission to do genomic sequencing, but of course that could be arranged. But this particular gene is quite common, so it would be no problem to find it in the population. Yeah.
And as Kathy Sheeper said last evening, confirmation of the allelic variants that she has inferred has been a very long and costly exercise. I think that one of the principal issues to bear in mind when we speak of inferences, we know from AIR data that there are so many unreported alleles out there. They're being found at a great rate, but they're not going to be confirmed. I mean, a handful will be confirmed, I suspect, but so many more will be discovered by people whose principal interest isn't in the discovery of genes. They're interested in analysing the data, and in the process, they see that there's a new polymorphism that they can infer, but they want to move on and get on with their main game. And so that's why we've put in place this process for the reporting of inferred sequences.
Yeah, one thing I could add to that, we have tried to do some germline confirmation, but once you start moving beyond one or two patients who have 2 or 3 novel alleles, you get up to tens or approaching hundreds of individuals, then the workload becomes enormous to confirm all the novel alleles. So what we've tended to go for is confirm a subset of alleles and just confirm a full genotype of one individual, just to show that technique works. So if we have a working technique with high confidence in the alleles it can detect, then you should be able to apply that to multiple individuals and not have to genomically verify every single one of the novel alleles.
Yeah, Scott.
The criteria that the germline working group has come up with, how does that, or how will that relate to parameters in these computer programs? Do we know what that relationship is? So to ask it another way, will users know what parameters they should or should not use, which won't, when they're set to values, are they out of bounds? And that means the inferences are not going to be accepted by this committee that's been suggested by and are there values that will be? Has that been thought about? What do we know about that relationship?
I think what I'd say is that the working group recognises that it's going to be a long time before we can articulate in great detail those kinds of issues, and that's one of the reasons why the proposal that we've put forward is that there be guidelines circulated so that people understand the kinds of issues that the working group believes should be addressed. But ultimately, if someone infers a new sequence using a process of inference that has been published, then the inferred sequence will be considered by the review committee. And the review committee will consider the sequence in the light of the general recommendations, but ultimately it will be their call, and the suggestion is that their approach should be conservative. I think everyone here would agree that there are calls we can all make where it's bleedingly obvious that you've got a polymorphism, and those are the kinds of inferences that we need to focus on in the first instance. The calling polymorphisms of rarely utilized sequences is going to be a much harder issue. It's going to be much harder for us to reach agreement, and we don't intend to try that in the short term.
I'm just interested in hearing the panel's view on validation through haplotype analysis by finding individuals that are heterozygotes for D segments or J segments, because we're sort of in the beginning of everything, and it's only now that we're starting to sequence hundreds of individuals as opposed to handfuls of individuals. So in the coming years, there will be thousands, presumably, of humans that will be sequenced by these methods and inferred germlines. So the number of people we find that are heterozygotes for Ds and Js will increase rapidly. And then, so as that increases, we'll also have an increased chance of validating novel Vs.
Yeah, I would say on the haplotyping issue that, one of our problems is that heterozygosity in the J locus, or J6, is generally at about 35% of the population. So there are large numbers of people for whom haplotyping using the J is unavailable. There might be heterozygosity at a D locus, but of course the D genes are utilized at lower frequency, and in fact some of the D genes that are polymorphic are utilized at particularly low frequencies, so they're not particularly useful. I think in the not too distant future, our great haplotype hope will come from the constant region, because as Mari Paul reported many decades ago, there's plenty of heterozygosity in the constant region genes, and if we have longer reads that can reach the points of difference between those constant region alleles, then we're going to be able to expand haplotyping, I would imagine, to the entire population.
I'd also add that I think it's going to be, important for the methods, as we generate those data, to have sort of public collections of paired, expressed repertoire sequencing with some kind of genomic validation. Is going to be important. Is going to really allow a lot of these methods to be further developed.
Do we have any more questions? I think not. Okay. Well, would you? Oh,
can I just make one comment, because I don't think there's a time to talk about it. We talked a lot about yesterday about software formats. So as many people see, there's a lot of tools that are being developed, multiple tools for inference of alleles, but also for things like cloning, VDJ assignment. And one of the things we talked about yesterday as a community, but I think there were not a lot of people there, is that one of the things we want to do is try and come up with some common file formats where people can exchange these kinds of data and run multiple different types of methods. So you can easily compare, say, IGDiscover and Tigger and other methods and sort of do plug and play. So for anyone who wasn't there yesterday who wants to talk about common file formats, who's writing tools, please talk to Uri, talk to me or anyone on those committees.
okay, so will you join me in thanking all of this morning's speakers? And now over to Felix.
I'm just going to take 2 minutes if I could. So just to remind people, on Wednesday morning, the community is going to be asked to vote on working group structure and plans and on the governance structure. We've gotten some feedback that we've had presentations about the working groups, but people would like to see kind of more formally and written out what these pieces are going to look like. So I'm going to be talking to the working groups about this, but we'll ask them to circulate by tomorrow afternoon and out suggestions for their leadership. How many co-leaders are they going to want to have, who these might be. 1 or 2 sentences on the purpose of the working group. We have a lot of that done already. And then kind of an overview for the plan for the coming year in bullet points. So we're going to try to get that out by tomorrow afternoon so people have something to kind of hold on to on Wednesday morning when they do that vote. And then Jamie talked about governance, and she's sending out I guess a Word file or something so that you can put comments in and send them back to her, and we'll try to get those incorporated into the governance structure that we'd be again voting on Wednesday morning. So we have a short break, and then we come back for germline haplotypes.